So yes, welcome everybody to the UK Freshwater Quality webinar. And myself and Joe Holden here at University of Leeds oversee this programme that's funded by NERC and DEFRA. And here's a few objectives, outlines the objectives of the programme to really strengthen our understanding of the sources, behaviour and fate of pollutants within our freshwater systems, to increase our knowledge of how pollutants are accumulating within our environment and how they have an impact on freshwater ecology. And really the outputs from this programme are to better inform policymakers and enable us to adapt and mitigate the risks of pollutants and climate change to our freshwater ecosystems. So that's a little bit about the programme. And within the programme, we have 5 research projects. And over the last 6 or 7 months, since January, I think, when it was Alistair and EcoMix kicked us off, we've heard from all of the projects listed here. And today, we're going to be hearing from the last of the projects. We've heard from all the others this year so far. And then we're going to hear today from the long-term large-scale freshwater ecosystems project. So if you want to get in contact with us, I think many of you probably already know about this, but just to put up some contact details, you can email us, freshwater@leeds.ac.uk. We have a website, which is can be found here. And we're also on X, and that's our Freshwater Qual 1, where you can follow us and hear about updates from the programme, but also other relevant information to do with freshwater ecosystems. So before I pass over to Vicky and the team here from the long-term large-scale freshwater ecosystems project, just to let you know that we will be stopping for summer break, and then we'll be resuming at the end of September with a webinar looking at co-contaminant exposure in the aquatic environment following antibiotic manufacturing in September. Then in October, we'll be hearing from Yorkshire Water, looking at chemical investigation programme. And then in November, we'll be hearing about biosecurity and invasive non-native species. So we've got some other talks lined up for you for the rest of the year. And then I'll just hand over to Vicky, Stephen and Ian, who's going to tell us a little bit about their progress in their project within the programme. So over to you guys.
Thanks, Pippa. Just opening up the window now. Hoping that's in presentation mode. Does that look okay?
Yep, that looks great.
Brilliant. Thanks, Pippa and Joe. And good afternoon, everybody, and welcome to our long-term large-scale freshwater ecosystems presentation. I'm Vicky Bell from CEH, and I will be joined in this presentation by Steve Lofts, also from CEH, and Ian Vaughan from Cardiff University. And we're going to give you an update on our project, long-term large-scale freshwater ecosystems. So I'll be starting off. I'll tell you a bit of an overview of the modelling and how we go about it. Steve will describe the all-important pollutants themselves, where they come from, and projected future changes. Ian will add the freshwater ecology to the modelling approach, and I'll come back at the end briefly to give you a summary of what the project will deliver. So, but before all of that, I'd like you to meet the project team. We are 5 organisations: UKCH, Rothamsted Research, Cardiff University, Boburn Consultancy and the British Geological Survey. There's a wide range of expertise ranging from hydrology, agriculture, metals, future scenarios, ecology, a huge amount of expertise. There's a list there and also a picture of our very first kick-off meeting in January 2023. And we're supported by our stakeholders. We have currently 11 stakeholders. I've put their logos there. But if anybody in the audience would like to be part of our team, do get in touch. So I'm going to give you a little bit of background, partly because, well, a number of people on this new project long-term large-scale freshwater ecology ecosystem, sorry, we worked together on a previous NERC macronutrients project called long-term large-scale. You'll note the similarity in the name, which ran from 2012 to 2015, led by Professor Ed Tipping. And in that project, we explored the past, specifically how carbon, nitrogen, and phosphorus in UK, well, across the UK, in the environment, particularly in my case, the river, changed since 1800. So it's going right back a couple of hundred years. And I was involved in the work that simulated freshwater macronutrients in UK rivers at the national scale. And we did this basically for the whole of the UK by focusing on the dominant sources and processes impacting carbon, nitrogen, and phosphorus in our environment, hence the name long-term and large scale. The little map on the bottom right shows the sort of outputs. This is what the outputs look like. I think this was probably a flux of nitrogen across the UK for a particular year. And you can see that it's all across the terrestrial part of the UK, and we are also with this able to provide marine outputs. So in the new project, we hope to go the other way. We're not looking back at the past anymore. We want to explore the future. We want to explore future projections of freshwater quality and biodiversity. And we're benefiting from very recently available data sets for climate, UK CP18, and socioeconomic scenarios. And we can use these to explore the future in a similar way to how we explored the past. So again, we will use our long-term and large-scale modelling approach to identify dominant drivers of change in our UK rivers and how they in turn will impact on our freshwater ecosystems. So I'm aware that models mean different things to different people and different scientific disciplines. So before I sort of dive in to tell you about the model, I just wanted to clarify what we mean by a model and why we're using one. Well, in this sort of context that we're using here, we're using a model summarises our understanding of the world, how it works, and usually we use mathematical equations, and in these models, computer code. But we know that the world is highly complex, and our models typically need to focus on the most important parts, and of course they need to make simplifications. So here's an example. Here we have a nice photo of Gorpley Clough Nature Reserve in Yorkshire. That's the reality. It's complex, a web of soils, land cover, trees, the water, going to be nutrients in the water, etc. For a model to represent that, it really means simplifying it down. If I was to try and model this environment with a model, we'd need to take account of land cover, how that might change in the past and the future. We'd need to take account of changing weather, often daily. That influences soil moisture, runoff, and perhaps in the future there might be changes to the pollutants entering the stream. And the model has to take account of all of this, and of course it needs to make simplifications. But models are useful. They can help us understand the past or the future without having to time travel, and they let us explore places we can't measure and even test scenarios. But it's important that we're grounded in reality and we check our model against observations to help us test and improve our understanding of the world. So how are we making this model? Well, as I said, we're basing what we're doing in this project on a lot of existing technology, which is brilliant. I started modelling rivers at the national scale when I was modelling river flows, and I was working on a grid-to-grid hydrological model, and that ran across the UK in a 1 kilometre resolution. But the NERC macronutrients LTLS project enabled me to work with experts in nutrients, lakes, geology, and other chemistry, and we managed to incorporate this information on a 5 kilometre grid across the UK. Now, this current project takes it one step further. We're able to bring in expertise in metals and micropollutants, brilliant new data sets, downscaled UK CP18 data, the climate data, and the new speed socioeconomic scenarios, which give us an idea of the possible range of futures that we might have. And Steve will tell you more about those in the next part of the presentation. And we're going to work with ecologists, such as Ian, to introduce freshwater ecology into our scheme. So what exactly are we modelling? Well, this lot, these are the freshwater physical variables we are modelling. Water, of course, I started doing that first, macronutrients in the LTLS project, But we're bringing in lots of new micropollutants, pharmaceuticals, polyaromatic hydrocarbons, pesticides, and a range of metals, including there you'll see copper and cadmium. And I mentioned those because they're coming up later in a couple of diagrams. And we're also modelling other water quality variables such as pH, temperature, oxygen, and many more. So what processes are we including? Well, may or may not be pleased to hear, I'm not going to go through the whole model. We don't have time for that, but what I'm going to do is give you a summary of the sort of things the freshwater model's thinking about, and I'll provide you with references at the end for more information. So the model itself is gridded. It runs for long periods. It takes account of changes in weather or climate for future scenarios. Rainfall lands in terrestrial models of semi-natural or agricultural landscapes. And these soil and vegetation models provide an estimate of pollutant runoff to our rivers. And the river model runs river flows, estimates runoff, lakes, urban areas, freshwater chemistry, groundwater pathways, and we're also now linking to a freshwater biology model. The future scenarios, which Steve will tell you a bit more about shortly, encompass lots of things that will happen to our landscape in the future. Climate projections, changes in land use, fertilizer use, grazing densities of animals, wastewater, and pollutants from acidity, metals, mines, micropollutants. And we're able to look at our outputs, and what we aim to get out of this is national scale scenario outputs of things like that list on the right. Water quality in lakes and rivers, gas fluxes, biological metrics, and community structure. There's a huge amount of information that this modelling approach can provide you, and there's going to be quite a big job to turn it into something that's useful. So, brief overview before I pass to Steve. Broadly, just to give you an understanding of the project as a whole, we're going to model concentrators of chemical stresses in rivers for the whole of the UK, both the present day, so we can compare against observations, and for the future for a number of scenarios. Then we want to examine what the biological response is to changing chemical stresses. And finally, we want to explore the best ways of presenting all of these outputs so they can be used by policymakers and decision makers, and also make our outputs available for follow-on researchers. And at that point, I'd very much like to pass to Steve. Over to you, Steve.
Thank you very much, Vicky, and good morning, everyone. Could we go to the next slide, please? Okay, so I'm going to tell you a little bit about how we're dealing with pollutant sources, both for the present day and for our future scenarios. So the first and very important thing to say is that, of course, different pollutants will have different sources into fresh waters depending on what they are and how they are used. So, for example, atmospheric deposition can be a source of metals, polyaromatic hydrocarbons and other industrial organic chemicals, acidifying contaminants and nutrients. Whereas, for example, the application of biosolids or animal manures to agricultural soils can be a source of nutrients, metals, potentially organic compounds, and also pharmaceutical compounds as well. And again, in agricultural soils, the use of pesticides is our main source of that class of compounds. So when we come to look at producing emissions of our chemicals, which of course are central to driving the model and its predictions, we need to think about how these particular how each particular chemical is used and therefore what its sources are going to be. Next slide, please. So just to put the consideration of where our emissions data are within the network of models that we have an atmospheric deposition model, we have soils models, groundwater model, and everything then runs into the freshwater models. And our emissions, according to substance and source type, may be to the atmosphere, they may be from agricultural activities, and they may be direct to surface waters. So our emissions do need to be broken down because of the different spatial pattern of where they occur and how they occur, and whether they go into the atmosphere and then are deposited into soils and then can be leached to surface waters or directly into surface waters. Next slide, please. So I wanted to jump straight into showing you some examples of the estimated present day emissions that we are doing. And this example is 4 different types of emissions for copper. This is not exhaustive. This is very much work in progress. But what you can see here is estimated emissions from 4 different sources of copper. Wet atmospheric deposition, copper in rainfall, so that has been done by estimation of emissions and running them through EMET4 UK, our atmospheric deposition model, in order to produce a present day map that you can see there. Domestic emissions to wastewater, service emissions, that really sort of means town centres, offices, etc. And urban runoff to wastewater are the other examples that you can see there. They are at the moment quantified in slightly different ways, but those will be integrated, and these will, if you like, form layers of the overall copper emissions ultimately into fresh waters. Next slide, please. So those are the present day emissions, but we're putting a big emphasis in this project on the future scenarios. And we're using a set of scenarios called the UK Shared Socioeconomic Pathways, which have been developed in previous work that CEH has been involved in. They are downscaling of global scenarios, which have really been developed in order to investigate the issues around climate change, adaptation and mitigation under different possible trajectories of the global future. So those have been brought down to the UK scale. There are 5 of them. There is a sustainability one. There is a very fossil fueled rich one. There is one centered around the development of a high degree of social inequality. And there is one centered around a real sort of cutting off of international trade and an attempt to become much more self-sufficient in terms of resources and economics. And there is also the middle of the road scenario, which, while not a business as usual scenario, is the scenario which shows the least change from the present day. And collectively, these scenarios are intended to present quite an extreme range of 5 possibilities from the infinite number of potential future trajectories for the UK. And the real idea about using these is that the issues that we will show in terms of water quality in our modelling of these scenarios can be traced back to particular drivers within the scenarios themselves. And then that can be put in the context of a policy challenge to stakeholders, which I think Vicky will talk about in a little bit. As I said, these scenarios have already been developed, and so our jumping off point at the start of this project was already quite a lot of information. Each scenario has a narrative, which is a prose description of its key events and trends. Then there are semi-quantitative trends in a large number of socioeconomic variables. The set of graphs you can see there on the right is the semi-quantitative trend in population under each scenario. And there are also a fairly large number of quantified projections, many of these spatial, and they critically, as I've highlighted there, include land use, which is really important for determining what emissions of a particular chemical go into which grid square, and also key variables such as population and healthcare, which, depending on the use of each chemical, will have varying importance in controlling what the future trends in that chemical are. And although the projections for the scenarios largely go to 2100, unfortunately, not all the key information that we need for modelling goes all the way to 2100. So we are modelling to 2080. But the projections that I'm going to show you will go to 2100 because that is the most convenient way that we can make those projections, just to think about the whole timescale of the SSPs. Next slide, please. So how do we come up with trends in chemical emissions under our future scenarios? Well, what I've put at the left here is really the sort of thought process, the steps in the process of doing that. So we start with a substance and we have to create a use profile for that substance. How is it used? And therefore, where and what are the patterns spatially of its emissions? And what I've shown you here, again, this is not exhaustive, is a simplified use profile table for copper showing its applications and how those link into the source type. So, for example, car brakes can contain a lot of copper. Car braking wears that copper off onto road surfaces where it can then get into the urban runoff. Copper can be added to animal feed in order to promote growth, particularly, for example, for pigs. So you then have copper in manure, and manure application to the land puts that copper into the soil. So after the use profile, we then look very, very closely at the source to freshwater pathways, which are very closely linked to the use profile as shown here. And then we come up with what we call proxy drivers for chemicals. So these are variables that directly drive the rate of emissions. And from those, what we are doing in this project is first looking to make those trends under each scenario in those proxy drivers semi-quantitatively, and then to do it quantitatively. So over on the far right-hand side, we have a semi-quantitative trend in a proxy driver that affects lots of chemicals. And that's really the efficiency and the capacity of the wastewater treatment system. How well will our wastewater treatment system function in the future? And this was one of 4 drivers which we explored with stakeholders at a workshop back in September. Our stakeholders had a very brave and really good crack at drawing what they thought would be semi-quantitative trends in this proxy up to 2100. We have reviewed those trends internally, and what you can see there is our final semi-quantitative trend for this particular proxy. But we do also have, from previous work under the SPEED project, we do already have quite a lot of quantitative trends that we came up with by a consensus approach with a subgroup of people from within the project. And what you can see in the middle at the bottom here is our projections for another key variable for chemicals in agricultural systems, the total number of cows within the UK. Obviously, the more cows there are, the more veterinary pharmaceuticals may be used, the more other chemicals they may get in their feed, and this all comes out in the manure, and some of it at least will then end up in the soil. So this is an example of a key driver where we already have a quantitative estimate of how we want our trends to go. And I think that's it from me. Thank you very much for listening. And we move on, I think, to Ian.
Yeah, thanks, Steve. So I'll come talk very briefly about the ecological components, and this will be necessarily brief, partly because I've got one eye on the time, but also because we're only just starting off with the ecological part. We had roughly sort of a year, just over a year's lag behind the rest of the project, so the physicochemical modelers could make a good start. So I said this will be a little bit more about what we're planning to do, but hopefully if we get invited back in, I don't know, a year's time, it'll be a bit more concrete. So next slide, please, Vicky. So just a little bit of background about the broader challenge of multiple stresses from an ecological perspective. And actually, I have a feeling Dan Reed may have presented something similar to this when he was talking about the Pacific project in one of the earlier seminars in this series. But broadly speaking, 3 general approaches we might take here, starting with experiments, often relatively small scale, very well controlled, getting that kind of real causal understanding of what's going on. The multiple stressor mix, more bespoke field investigations at slightly larger scale, perhaps longer timeframes, going out to the field to measure specific biodiversity variables or ecosystem processes like organic matter decomposition, that sort of thing. And then sort of the largest scales, sort of what you might term repurposing existing data. So national data sets often collected by the statutory agencies. And I know that across the Freshwater Quality Programme as a whole, all of those approaches are being used, and indeed some of the individual projects are using all 3. But we're nailing our colours to the mast and just working with the national scale data sets. And hopefully within that, we'll be looking across the range of different taxa that are commonly used for monitoring rivers. So in the picture on the right-hand side, look, diatoms, macrophytes, macroinvertebrates, and hopefully fish. So next slide, please, Vicky. So of course, with any of these approaches, there are strengths and weaknesses. In using large scale, these sort of national data sets, of course, we're trying to repurpose data which were collected for other purposes. So, there are particular challenges around that. The advantages in blue on the left here, we're actually going out into the environment. We're looking at the kind of the real mix of stresses, this complicated cocktail of different chemicals and other stresses that organisms are exposed to. By looking across the country, we can look at long stress gradients. So from rivers which are relatively clean water all the way to the most polluted locations, and that kind of real mix of different stresses everywhere in between. And of course, the other big thing is spatial and temporal coverage. Some groups, in at least some of the UK countries, we're looking at hundreds, maybe thousands of locations that have been sampled, often with sort of time series. We've got replication. So I guess sort of ideal kind of big data type conditions. Also, you could perhaps argue that by working at the national scale, there are particularly policy relevant things there too. Of course, the flip side for all those strengths are the weaknesses, the fact that the data weren't collected for this purpose. They represent a particular set of things that are recorded for typically statutory monitoring. So we're not getting some of the more detailed measurements around the biota. We're not measuring ecosystem processes. Coverage is non-random. And there are often big gaps in coverage as well. So for all this kind of many locations, many years, especially if we want to start connecting biological data to, say, water quality data that's been collected through monitoring, there are often quite big gaps. Different recording schemes recorded at different frequencies, different sites recorded for different purposes. And in some ways, that's actually held back this use of national data sets to some extent. The fact that quite often we may have biological data, but there aren't nearby water quality data, for instance. And then, of course, the last decade or so, we've seen big declines in coverage as well. So, great potential here, but equally all sorts of limitations. Next slide, please, Vicky. So these data have really been used in 2 ways. And the big sort of growth area in the last sort of few years, particularly the last sort of 3 to 4 years, has been sort of revisiting these data to get a more detailed overview of how status has changed. And of course, the statutory agencies were collecting data for this purpose originally, but various people have taken it on and gone into more detail. And here are just a few of the most recent papers that have come out in this area, everything from looking at data sets collected by agencies across Europe in the Nature paper at the top, the ones looking at England or England and Wales. And so from sort of a biological perspective, ecological perspective, we're seeing and have seen quite large improvements in quality. So in the 2 sort of centre panels there, you're seeing increases through time from roughly sort of 1990 towards the present day in things such as macroinvertebrate richness as a sort of general indicator of improving quality. The panel below that is looking at the proportion of sort of pollution-sensitive taxa, and that's thinking in terms of some of the more traditional forms of pollution, things such as raw sewage, that high organic matter loading and oxygen depletion. So generally, at least up until recent years, from 1990 onwards, a relatively positive story. More recent work, some of the stuff in the right-hand panel, this is work that's just been accepted in the last couple of weeks, is starting to kind of disaggregate that and identify particular areas where quality seems to be declining. And I'm afraid it's not a particularly large representation, but for example, in the lower panel there, that's showing a sort of a general decline, some sort of loss of these pollution-sensitive taxa from rivers draining the uplands of England and Wales. So it allows us to sort of say quite a lot about how sort of overall ecological status and conditions been changing? But making that link to drivers is a real challenge. So next slide, please, Vicky. So I hope is that this is where LTLS, once we plug in the FE component, can contribute. So from what Vicky and Steve have described, you can see that we're hoping to expand the range of stresses which we often look at with these national data. Can't remember which of it is. I think it's Vicky who showed the slide with the range of different chemical determinants we're going to look at. And of course, there are broad things around climate as well. By taking this modelling approach, so making predictions on this 5-kilometre grid through time, we have complete sort of spatial and temporal coverage for these stresses, hopefully, which then means, of course, we can plug in a lot of these gaps. So we don't have that problem of, well, we've got biological data from this location, but we don't have any nearby water quality monitoring data. So hopefully we can sort of extract more value from existing data resources. We're hoping to work UK-wide, so we're in the process of gathering data from all of the sort of 4 devolved nations. And then the big thing we're hoping to contribute to the wider Freshwater Quality Programme are these biodiversity forecasts under the different scenarios that Steve's described. So next slide, please, Vicky. One final figure just to give you a sense of what that might look like. So down in the bottom left, so my sort of box in the bottom left here, we'll be taking these sort of monitoring data, whether they're for macroinvertebrates or for fish or macrophytes or diatoms, and we'll be modelling those against sort of concurrent outputs from LTLS. So concentrations of different pollutants, nutrients, other things are from around sort of the same kind of time period as the biological data were collected. Once we've built those models and we've tested them, we'll then be able to apply them to the sort of the scenario outputs from the LTLS modelling, or indeed to look at time series as well, looking at, Steve was talking about up to 2080 or 2100. Potentially we could look at time steps on that journey as well. So that's my kind of box in the upper left here. I've just suggested perhaps a couple of pollutants here for space on a single slide. But again, you can see how you can marry the model to these scenario predictions to then generate our biodiversity predictions. So that's what we've got on the right-hand side here. So this is just, for example, 3 scenarios and just some sort of arbitrary idea of what outputs might look like. The key message here, as I say, hopefully if we come back again in a year or two's time, we'll be able to actually talk about this in concrete terms, is we're planning to generate predictions at the level of individual taxa. So that might be individual species, it might be the presence, absence, or probability of occurrence of different species within 5-kilometre grid squares, it might be their abundance, those sorts of things. In doing that, for a whole range of species, we're hoping to replicate the kind of data that would be collected if you went out and used a standardized sampling method in the field. So went out and did a 3-minute kick sample for macroinvertebrates, for example. By doing the prediction at the level of individual taxa, we can then build that up. We can sort of calculate community properties, things like species richness, the proportion of pollution-sensitive taxa, a whole host of different things. And then ultimately, we can work with the kind of biomonitoring metrics which are familiar to many people. So things like the trophic diatom index, the BNWP score, well, the WIT scores these days, life scores, and a whole range of other metrics to make our outputs as relevant as possible. So that's all I want to talk about in terms of the ecological component. And I know Vicky will talk a little bit more about outputs from the project as a whole in the final few slides. So back over to you, Vicky.
Thanks, Ian. I'm delighted to be able to, well, we haven't obviously we've got a couple more years to go on this project, but I'd very much like to tell you about the sort of outputs we're hoping to get from this project and even show you a sort of a prototype output we've just created. So our goal is to provide lots of information from all this modelling to support adaptation and mitigation of risks to freshwater quality across the UK. We want to make all the information we provide useful in as many ways as possible. So, for example, for stakeholders, we're keen to work with different stakeholders to try and derive information from our scenario outputs to help develop plans for adaptation, mitigation and detection of risks to water quality at a range of scales. So we'd like to talk to them about how this information we're providing can be best summarised and ways with different indicators we can use that we'll find useful. For other researchers, we'd like other people to use our work after we finish this project, and we want to share our models on GitHub. We're going to publish as many data sets as we can for use in follow-on projects, and of course we'll definitely publish our research in referee journal publications. But for others, we already have the project website. That's that red link there. And we're also planning to develop a web dissemination tool to allow people to zoom into that. Those UK maps are quite small and fiddly, but the web tool will enable people to zoom into their favoured part of the country and explore freshwater futures in nearby them or for an area of their interest. And that'll be made by our fabulous Gemma. So, where have you got to with all this? Well, over the past year, we've been working really hard to build a working prototype. This was always our aim. We wanted a prototype within the first year. I think we've just about done that. And we're just starting to run, see how the scenarios will work. And I'm delighted to be able to show you some really early outputs. These are by no means final outputs. In fact, we only made them a couple of days ago and they need checking, but it'll give you an idea of what that whole process is in action and what we hope to get out of this. So what I'm going to show you here is prototype future scenarios of how cadmium might change in the future. And what we're doing here is we're comparing cadmium around about the year 2000 or cadmium in about 2050, so about 50 years' time. And to understand what's happening in 50 years' time, we're exploring two different scenarios. Both of them are two of the scenarios that Steve told you about earlier. One is the sustainability or green future, SSP1, and the other is the more fossil-fueled future, SSP5, which is a more extreme future with expected to have greater climate change. So what we're looking at here, we're just focusing on one source of cadmium, that's atmospheric deposition of cadmium onto the environment. And you can immediately see we've got two maps here. The upper map is bright blue and the lower map is more of a red colour. And what these maps, the colours are showing percentage change in cadmium deposition between 2000 and 2050. So where it's blue, cadmium's gone down, and you can see that for the green future. And where it's gone red, it's gone up. And if it had stayed the same, it'd be sort of a grey colour. You'll see it's a bit patchy, but broadly, median across the whole of Britain, there is about 50% decrease for the green future and about 50% increase for the fossil fuel future. But the question is, what impact will that have on our rivers? Note here, we're not just changing a socio-economic scenario, we're also running through what the climate will look like in 2050. And for each of our futures, the green future and the fossil fuel future, we're using different climate model data because we're using a different representative concentration pathway, the more extreme one in the fossil fueled future. So we're combining two sources of information here, change in the input from cadmium and also change in the amount of water in our rivers. So these two fiddly red and blue maps are the impact on river cadmium concentrations. And again, these are maps of change between 2000 and 2050. And again, blue is a decrease in cadmium in our waters and red is an increase. Now, they're a bit more patchy than the deposition maps, and that's because we're combining both the climate and the cadmium emission. But I hope that if you squint and stay back from your screen, you'll see that the bottom map is rather redder, more red than the upper map, showing that for parts of the country in a fossil fuel future, we'd have quite an increase in river cadmium, whereas for the green future, there'd be more of a decrease, particularly in the south and east. And this is sort of information we could summarise in lots of different ways. We could summarise it regionally, or for particular rivers or water regions. Actually, here we've just gone for a simple approach. We're summarising these results in a simple bar chart. The first column shows median change all across Britain in the green future. The yellow bar shows a cadmium deposition decrease, and the purple is also a river cadmium decrease, but much lower. The fossil fueled future, the sort of right on the right, it goes the opposite way. We have more of an increase in cadmium deposition and also an increase in cadmium in the rivers. So broadly, Freshwater cadmium, according to these prototype results, decreases in the green future and would increase in the fossil fuel scenario. But please note, these are not final results. They are just a prototype. Okay, so what will these scenarios provide? All of this work, we hope to provide a number of scenarios for stakeholders to use. We're looking at change in biodiversity, change in overall maps of change across the country, like you've just seen, and also future trends of pollutants at particular sites. How can we support decision making? Well, running a model allows us to add and remove bits of the world. For example, we can remove pollutants from different sources and freshwater processes to understand where the pollutants come from and their fate. Let me show you what I mean. So this graph here is actually historical. It's not the future, and it shows time along the bottom is about 200 years. The bars are individual years, and up the side we have nitrogen in the rivers in kilotons per year. The overall trend in the 19th and 20th century is upwards, and then after that there's a bit of a decrease. But if you look at the colours, individual colours in the bars, we've turned off parts of the model to understand where the nitrogen comes from. We can turn off the different sources one by one to understand how much of the nitrogen in our rivers comes from different places. And this is across the UK as a whole. And we can also look at within our rivers, where does all the nitrogen go? Does it go to the coast, or does some get stored in groundwater and released later? So this is quite a powerful tool that a model can help you determine where your nutrients come from and where do they go. So we look forward to discussing these sort of scenario outcomes with our stakeholders, and we hope to understand how best to present all these results to support policymakers. Lots of different ways to do this. One way we're thinking of is called a driver pressure state impact response framework, and this sort of approach can provide a useful structure for assessing causal links from socioeconomic factors, driving pollutant releases through to human impacts. And we'd like to work with stakeholders to explore how these results and the future scenarios could impact on policies and further positive development. So next steps, we're still in the model development phase. It's only in the second year of our project. We want to finish our model, test against observations, and continue the development of future scenarios. We're keen to start delivering some outputs to our freshwater ecological modelers to develop understanding of the impact on freshwater biology, and we want to work with our stakeholders to understand how best to present and provide the future scenarios. Thank you very much. We welcome questions, and for those of you who'd like to know more about the project or any of the models and tools, there are some links there. Thank you.